Okay, that means we're ready to move on to the next set of lightning talks. I'd ask that those of you who are giving the lightning talks in person maybe take seat in the front row and to be ready to jump on stage when it's your turn. A couple of them will be presented from online, however, and that is actually also the first case, the case for the first talk by Matt Coelho from the Wellcome Sanger Institute. And I think he's joining us remotely over Zoom, and he's already ready. Well,
yeah, hi, thanks very much.
Thank you.
Okay, great. Yeah, thanks very much for the invitation. I'm Matt Coelho from the Wellcome Sanger Institute in Cambridge, UK, working in the laboratory of Dr. Garnett. And today I'll be talking to you about the application of base editors to perform mutational screens in cancer cell lines. Today we're focusing on the interferon gamma pathway. And interferon gamma pathway, because variants can be causal in many different human diseases. Many of these are missense variants, for example, with unknown clinical significance. These can lead to hereditary immunodeficiencies, but also inflammatory diseases. And somatic mutations can also lead to myeloproliferative neoplasms and also leukemias. We're really interested, though, in the role in anti-tumour immunity, because this is increasingly relevant therapeutically with anti-tumour immune checkpoint blockade inhibitors, which are used for many different cancers now in the clinic. And these work by, if you look on the left, activating this JAK-STAT signaling pathway in the tumour cell when there's, for example, interferon gamma, this cytokine in the tumour microenvironment. And this leads to tumour cell death normally, or cell cycle arrest. However, when these loss of function variants occur in the cancer cells, they no longer die, and this can lead to therapy resistance or evasion of the immune system. So to actually define what these variants were doing throughout the whole pathway and see if they're loss of function, we used the modality of base editing, which is applicable in this case because we needed to work in a cell line which was responsive to interferon gamma, and a cancer cell line, for example, in colorectal cancer. Many of these have triploid genomes or more. So the cell line we're using is triploid, and we wanted to analyze the variant function in the endogenous locus. So as Holly described from Beam Therapeutics, we can use these wonderful base editing tools. For example, the cytidine and the adenine base editors together to increase the number of mutations we can install in the endogenous locus throughout the interferon gamma pathway. And we're using Cas9 variants with increased, reduced PAM specificity, so the NGN variants, to get more variants throughout the genes. And for example, this is our experimental setup. So essentially we select with interferon gamma in the dish, and then we have proliferation or FACS readouts based on response to this cytokine. Okay, so I don't have time to go through all the data in this short presentation, but if we were to pool the adenine and cytidine mutational scans together using base editing across, for example, JAK1, which is a kinase downstream of the receptor, we find that each dot here is a guide RNA. And then the positive scores here mean that they're enriched, so they're loss of function, and the negative ones, which are rarer, are gain of function, so sensitizing to interferon gamma, killing the tumor cells more rapidly. And what's really exciting is that some of these missense variants have been found clinically in cancer, in other diseases, but also in patients which have failed to respond to some immunotherapies, which means there might be some clinical relevance here. And how did the mechanism of action work for some of these missense variants, which we've defined? So for example, some of the blue ones in JAK1, loss of function, and they're surrounding the kinase active site, and ADP and magnesium. And interestingly, we found a gain of function site here in red, which actually disrupts the protein-protein interaction interface, which is known with the negative regulator SOX1. So when we want to validate these loss of function and gain of function mutations, we went to for example, one of the things we did was amplicon sequencing, because essentially we're predicting the edit from the guide RNA sequence alone in most cases. And we wanted to see how accurate that was in the endogenous locus by sequencing. And that's what's shown here. So in orange is the remarkably good editing efficiency we're getting around 40 to 50% in the cells. And in red is the frequency of the edit with interferon gamma selection. So this is showing that many of these loss of function mutations in blue are actually enriched, so they're loss of function bona fide mutations. And this is the way we can validate the specificity and also the actual edit that we're getting in the locus, which very, very well correlates to our predictions. Another advantage of base editing, although it can't make all of the mutations like SGE, we can use it in many different cell models, challenging cell models, for example, in tumor organoids, where we have autologous anti-tumor T cells from the same patient. And we repeated the base editing mutagenesis screen in a 3D organoid and found actually that we could recapitulate lots of the 2D cancer cell line data as well, based on the loss of function and gain of function mutations throughout the pathway. And finally, we did some validation in the co-cultures, because these anti-tumor T cells would be expected not to be able to kill the loss of function JAK1 mutations in these cells harboring the JAK1 mutations in the tumor organoids, and the gain of function organoids should be more sensitive to T cells. And that's what we saw. It's quantified here. So in blue are the loss of function missense mutations. And what's really exciting is in red is the gain of function missense mutations, which are more sensitive to anti-tumor T cell attack, which has therapeutic implications, we think. So just to wrap up, this is also summarized in a poster and also a preprint we have online at the moment. So we've essentially mapped many putative mutations throughout the pathway and done a lot of validation work to back this up. So yeah, if you're interested, please link up with the poster or the bioRxiv preprint or contact me afterwards. So thanks very much, and welcome any questions.
Thank you, Matt. We're running a little bit over time, so maybe we have room for one question. I see we have a question online.
Yeah, so William Bushner asks, how do you do all the NGS verification? Was it high throughput in some way?
Yeah, so the NGS verification, essentially we've been doing amplicon sequencing across as many guys as we could, which is not terribly high throughput, but that was kind of like benchmarking our prediction of the edits. And more recently, we've got into single cell DNA sequencing type methods, which are more high throughput. So we can do it kind of en masse without doing multiplex amplicon sequencing.
Okay, thank you again, Matt.